Hello, and welcome to Echoes of the Forgotten Earth, a corner of the universe where curiosity kicks off its shoes, settles into the couch, and starts wondering about everything that existed long before we did. Tonight, we're slipping backwards. Not a handful of centuries or a few scattered millennia, not even a mere million years. We're heading billions of years into the past, into an era with no photographs, no fossils you'd recognize at a glance, and no comforting familiarity of modern maps. A time when the land masses bore almost no resemblance to today's continents, when the air would have felt alien to us, and life, if you'd even call it that at this stage, was mostly invisible, quietly occupied with making some of the most consequential choices in the planet's entire story. Tonight, we're stepping into the Proterozoic Eon. Maybe that term has never crossed your path before. That's fine. It doesn't come up much in small talk. It rarely headlines museum exhibits, and it doesn't wedge itself into school syllabi the way dinosaurs or mass extinctions do. Yet the Proterozoic is enormous, almost beyond comprehension. It stretches from roughly 2.5 billion years ago all the way to about 541 million years ago. More than half of Earth's entire history sits folded inside that interval. So if you lined up everything that has ever happened from trilobites to tyrannosaurs to tweets, the Proterozoic would be the long, hushed overture. But it was not boring, not at all. This was an age when Earth's outer skin was reworked again and again. Continents smashed together and tore apart. The sky slowly filled with something entirely new and mildly poisonous to many early inhabitants: oxygen. Microscopic organisms, barely more than specks, stumbled upon photosynthesis without asking permission from anyone. Cells joined forces, fused with other cells, and unintentionally laid down the groundwork for everything that would follow, including us. The rocks that formed in this era don't easily yield their stories. They murmur, if they say anything at all. But tucked away in those ancient layers are records of the first great global ice ages, the first truly complex cells, the first attempts at cooperation, the earliest signs that life was not satisfied with staying tiny and uncomplicated. If the Archean Eon was Earth's unruly, chaotic childhood, the Proterozoic was its drawn-out adolescence. Awkward, intense, and full of irreversible transformations. So if you're ready to drift backward, let's slip into an age when the sun shone dimmer, the moon sat closer, and a full day had not yet settled on 24 hours. A time before animals, before plants, before the world had a word for what it meant to be alive. Back when Earth was still working out who it wanted to be. No hurry. We've got billions of years in front of us, and it all begins with a planet just starting to wake. At the dawn of the Proterozoic, Earth was already more than 1 billion years old. It had been pounded by meteor impacts, smothered in massive volcanic outpourings, and wrapped in a hazy shroud of gases that would be lethal to us today. There were oceans, certainly, but not the clear blue expanses we picture now. They were cloudy, metallic, and anoxic, completely devoid of free oxygen. In those primeval waters, iron drifted freely, dissolved in great quantities, tinting some stretches of sea a faint reddish hue. What little land had managed to rise above the waves was naked rock. No forests, no soil, no chirping insects, no rustling foliage. Only wind, silence, and the patient, relentless workings of chemistry. This was not a lifeless planet, but it was subdued. The kind of stillness that settles over a world where life is tiny, slow, and entirely occupied with vital internal business. Single-celled organisms already flourished. They had been thriving for a very long time. Ancient varieties of bacteria and archaea had already mastered survival in brutal environments boiling hydrothermal vents, hypersaline pools, dark places soaked in sulfur. They were not glamorous, but they were astonishingly efficient. They broke down minerals. They took in and released gases. They built structures far too small for any unaided eye to detect. And in doing all this, they quietly began reshaping the entire planet from the inside out. Continents were just beginning to organize themselves, though nothing about them would have looked recognizable to us. Early supercontinents with names like Columbia or Nuna, if you care to go hunting through geology papers, were assembling. They drifted ponderously over the mantle, steered by convection currents roiling in Earth's molten interior. Plate tectonics was already in operation, but it hadn't yet sculpted the spectacular mountain ranges or deep ocean trenches that decorate modern maps. Instead, landmasses sat low, fairly flat and heavily volcanic. Erosion had not yet piled up thick blankets of sediment, and without plants to hold soil in place, any loose material was at the mercy of rain and wind. Rivers existed, but their behavior would have been unfamiliar. Lakes pooled in impact craters, or in stretched-out fractures where the crust had begun to pull apart. Everything felt raw, unbuffered. The elements operated almost directly on rock and water without the interference of sprawling, complex ecosystems. Sunlight did reach Earth, of course, but it was weaker than what we live under now. The young sun, still settling into its main sequence adulthood, radiated only about 85% of the power it sends us today. That reduced energy forced the planet to cling more carefully to what warmth it had, and it managed in part thanks to potent greenhouse gases like methane and carbon dioxide. The air was thick with them. There was no ozone shield yet, so harsh ultraviolet radiation streamed in from space, making the surface a perilous environment for anything delicate. Life remained mostly in the oceans, sheltering beneath layers of water that acted as a natural shield from that radiation. The deeper zones were safer, calmer, and far more stable. All the while, the crust below continued its slow rearrangement. Early versions of continents had already started forming in the Archean, but during the Proterozoic, the growth of cratons accelerated. Cratons are the ancient, stable cores of continents, the geological elders beneath your feet. Once these continental hearts form, they tend to endure. Thick, buoyant, and resistant to deformation, they become the skeleton of continents to come. Much of present-day North America rests on a craton that locked into place during this time. The same goes for large portions of Africa and Australia. Fragments of these ancient blocks have survived billions of years of collision, rifting, erosion, and volcanic upheaval. They are remnants of a world still learning how to keep its surface in one piece. One of the quieter yet profound transformations of this eon unfolded in the mantle. Radioactive elements decaying deep within Earth were a major source of internal heat, and over long stretches of time that heat drove cycles of melting, cooling, and re-melting. These processes birthed new types of rock. Granite became more widespread. So did certain minerals that would later prove crucial for life, phosphorus, for example, and molybdenum. These elements had no idea they would someday power enzymes or help build DNA, but their presence and distribution would quietly constrain and enable biological possibilities for the rest of time. Meanwhile, in the oceans, something unprecedented was beginning to stir. Among countless microbes, a particular group had begun to perfect a difficult and risky way of harvesting energy. They were bacteria, but unlike the sulfur-loving or methane-producing species that came before them, they had learned to exploit light, not just any light, but the full, unfiltered sunlight streaming into shallow seas. With that light, they discovered a way to split water molecules and siphon off energy. As a side effect, they released something unusual into their surroundings: oxygen. This seemingly minor waste product would grow into one of the most disruptive forces in Earth's history. But right now, it was only beginning. Tiny bubbles of oxygen, unseen and seemingly unimportant, drifted through waters that had no inkling of what they were heralding. The world, in a sense, was still half asleep. But someone had just cracked a window open, and soon the atmosphere would be transformed beyond recognition. The seas of the early Proterozoic were nothing like our modern oceans. Yes, there was water, and plenty of it. Vast basins draped across much of the globe, as there are now. But their chemistry, their colors, their textures belonged to another planet's story entirely. These were anoxic oceans, stripped of any free dissolved oxygen. And that single fact changed everything. Without oxygen, Many of the biological pathways we now consider standard simply could not operate. The kind of respiration that fuels most organisms today hadn't evolved in any dominant way. Instead, microbial life depended on alternative strategies, ones that might seem almost alien from our perspective. They tapped into sulfur compounds. They produced methane. They wielded iron and nitrogen in ways that feel more like unusual lab reactions than survival tactics. Those methods worked, but they were slow, and the payoff was limited. These were ecosystems founded not on combustion like respiration, but on fermentation and low-yield chemistry. The very hue of the water likely shifted from place to place, from murky green to muddy red. Iron permeated everything, existing freely in dissolved form. In modern oceans, iron is scarce. It quickly reacts with oxygen and precipitates out. But back then, with no oxygen around to spark that reaction, iron could drift for long periods throughout the water column until something triggered it to drop. Often, that something was the waste oxygen leaking from newly evolved photosynthetic microbes. When that oxygen met iron, a chemical reaction followed, and the iron oxidized or rusted, then fell from solution in vast, settling sheets. From this, banded iron formations were born layered sedimentary rocks that are still heavily mined today. They accumulated over hundreds of millions of years, laid down in alternating sequences of iron-rich and iron-poor deposits. Each band is a record, a thin, stony diary entry about changing chemical equilibriums, about rises and dips in microbial productivity, about a planet in the midst of a huge chemical transition. These formations are some of the finest archives we have for understanding Proterozoic oceans, and they require exactly the kind of iron-laden, oxygen-starved environments that dominated this time. Life thrived in these primitive seas, but you wouldn't have seen it with the naked eye. There were no fish gliding in schools, no seaweed forests, no corals, no crabs, not even plankton in the form we recognize today. Life was almost entirely microbial, living in films, layers, and mats. If you could zoom in closely enough, you might catch sight of stromatolites' layered mounds built by colonies of cyanobacteria and their microbial associates. These structures grew slowly, layer upon layer, as the microbes trapped and bound sediments, gradually forming mineralized domes. Some stromatolites grew in shallow tidal zones, others in deeper, calmer waters. Many were later buried and preserved as fossils, though a few modern examples persist in isolated parts of the world. These microbial mats were far more than carpets of identical cells. They were full-fledged ecosystems, intricate, vertical communities where different bacterial groups occupied distinct zones depending on light intensity and available chemicals. The upper layers captured sunlight and performed photosynthesis. The deeper layers devoured the byproducts and recycled nutrients. It was a vertically stacked, self-sustaining biosphere compressed into just a few centimeters of goo and sediment. Farther down, in the dim and lightless reaches of the sea, a different cast of microbes prevailed. Archaea and bacteria adapted to energy-poor conditions lived near hydrothermal vents or buried within sediments. Their metabolisms were sluggish, their lifespans measured in days or weeks instead of hours. But they were steady, and they played a crucial role in maintaining the ocean's chemical balance. With no large animals to churn through the seabed, the ocean floor remained a dark, motionless realm where chemical gradients could stretch out and evolve over immense spans of time. Methane was a dominant atmospheric character during this era. Certain microbes, called methanogens, produced it as a routine metabolic waste, releasing it into both the ocean and the air. Methane is a highly effective greenhouse gas, and its abundance likely helped keep the planet warm under the dimmer sun. For much of the Proterozoic, the atmosphere may have been composed mainly of nitrogen, carbon dioxide, water vapor, and methane, with only tiny, nearly undetectable traces of oxygen. The interplay between these gases and the chemistry of the oceans was delicate. Even small shifts in microbial processes could tilt the whole atmospheric composition. And once photosynthesis really ramped up its oxygen output, that chemical balance began to tip. Not with fireworks, not overnight, but gradually across millions of years. the scales started to move. Oxygen reacted rapidly with dissolved iron, with methane, with sulfur compounds. It vanished as quickly as microbes produced it. But the producers kept at it. They didn't need foresight. They didn't need to predict outcomes. They just ran their biochemical routines, splitting water, catching light, exhaling oxygen, and slowly, the oceans began to rust. This sweeping rusting did not proceed evenly across the globe. Shallow coastal waters near microbial mats felt the earliest changes. The deep ocean basins remained anoxic for far longer. Scientists describe this as a stratified ocean oxygenated or oxygen-tinged at the surface, oxygen-free below. Over time, as oxygen levels inched upward and began to penetrate deeper waters, the ocean's chemistry evolved. Sulfur took on a more prominent role. Hydrogen sulfide gas, generated by microbes thriving in low oxygen settings, accumulated in mid-depth zones. This created bodies of water that were both oxygen-free and rich in sulfide, fatal to most modern organisms, but ideal for many microbes of the time. Some researchers refer to these conditions as euxinic. They were toxic, certainly, but they were also stable, and they may have persisted off and on for hundreds of millions of years. It's a reminder that habitable is not a fixed concept. What would be instantly lethal to us was ordinary back then. What we categorize as extreme environments today were simply the baseline for much of Earth's early story. Pre-oxygen oceans were quiet to human senses, chemically intricate, and biologically rich in subtle ways. They sustained life, but life that played by a very different set of rules. These waters were sites of slow, profound transformations, silent revolutions in metabolism and energy use, and they formed the stage on which one of Earth's greatest upheavals was about to unfold. Because eventually, those tiny oxygen bubbles began to accumulate. It didn't begin with a dramatic crash. There was no single day when the oceans abruptly changed color or the sky suddenly shifted its tone. The great rusting of the sea's one name for this drawn-out transition was slow, soundless, and absolutely world-altering. When oxygen first began to appear in measurable amounts, it functioned more like a contaminant than a resource. The single-celled organisms that produced it, chiefly cyanobacteria, weren't doing the planet a favor. They were just running a more efficient energy harvesting method by capturing sunlight and splitting water. Biologically, it was an elegant innovation. Chemically, it was explosive in its consequences. Free oxygen is fiercely reactive. It doesn't sit around passively. It seeks out partners, especially metals like iron. And there was a vast store of dissolved iron in those ancient seas, lending the waters their greenish or brownish tint, depending on local conditions. With no oxygen around previously, that iron had nowhere else to go. Now, as small but growing amounts of oxygen began to persist, the chemistry shifted. Each time an oxygen molecule encountered an iron ion, a reaction followed. The iron oxidized, effectively rusting not in the flaky, crusty way we see on metal fences, but as tiny suspended particles. Those particles clumped, sank, and settled onto the seafloor. Over immense spans of time, they stacked up into thick layers of iron-rich sediment. These deposits hardened into rock. The result was the banded iron formations we see now, sheet-like rock sequences where iron-dense bands alternate with lighter, more silica-rich layers. They're scattered across nearly every continent. Thick deposits in places like Australia, Canada, South Africa, and Brazil. They differ in thickness, hue, and mineral makeup, but they all point toward the same story. A long-running chemical reconfiguration of the ancient oceans. This didn't proceed in a smooth, even line. It came in surges and lulls times when oxygen production boomed, followed by quieter intervals. Sometimes the oxygen spread far enough to oxidize immense swaths of seawater. At other times, it remained constrained near the mats that produced it. Earth was learning to breathe in irregular bursts. This iron-rusting phase likely stretched over hundreds of millions of years, and it wasn't just a curious mineral event. It marked the start of a cascading series of changes. As more and more iron was stripped out of solution, The oceans became chemically depleted in certain ways that pushed the system toward other transformations. Oxygen began to last longer in surface waters. Enough of it eventually escaped into the atmosphere. Methane, once a key greenhouse gas, began to react with this oxygen and break down. That reaction cooled the climate, a detail that would matter later. Even early on, free oxygen created entirely new biological pressures. For many anaerobic organisms, microbes that had evolved in oxygen-free conditions, this new gas was poisonous. It damaged delicate enzymes and interfered with basic metabolic processes. For a while, large sectors of the biosphere were effectively poisoned by the same gas that sustains us now. Whole branches of life may have vanished, erased silently by chemistry they were unable to tolerate. Others adapted. Some microbes evolved enzymes to neutralize oxygen's more harmful byproducts. Others retreated deeper underground or further into sediments, away from both sunlight and increasing oxygen levels. Evolution never stopped. It adjusted. Rocks preserve hints of this story. In places where banded iron deposits abruptly cease, where iron-rich layers thin out or disappear, we see evidence that the oceans had finally run out of iron to oxidize. Oxygen had removed most of the available iron, leaving behind waters that could now support an atmosphere starting to accumulate this reactive new gas in earnest. This prolonged shift, known as the Great Oxygenation Event, wasn't a one-night upheaval. It was more like a many-act play filled with long pauses, reversals, and subtle changes of direction. Yet it signaled a fundamental turning point. Earth's geochemistry had been altered at its core. The ocean shifted from iron-heavy and anoxic to something fundamentally new, an environment capable of supporting different forms of life, and eventually, genuine complexity. The ocean's color likely changed as well. Without iron to stain them greenish-brown, many regions of water probably cleared. As even modest levels of oxygen accumulated, new microbial groups rose to dominance. Light pierced deeper. New ecological layers formed. From a distance, Earth might have still appeared familiar. Blue seas, gray land, white clouds. But inside, everything was in motion. The rusting of the seas wasn't simply about minerals. It was about momentum, a change that once underway would be very hard to reverse. Oxygen had established a foothold. It was not going to disappear. The immediate consequences would be turbulent. As atmospheric oxygen rose, methane fell, greenhouse warming weakened, and the planet edged toward its first major deep freeze. But for the cyanobacteria in shallow waters, nothing seemed amiss. They had crafted a world in their own chemical image, whether any future creatures appreciated it or not. And somewhere in some quiet pond or coastal shallows, the oxygen kept bubbling to the surface. It's easy to miss the power of something so minute. Cyanobacteria are microscopic, just chains and loops of tiny cells, drifting or clinging to rock surfaces in places we rarely notice. Yet what they lacked in bulk, they more than made up for in impact. In the early Proterozoic, they were among the most consequential organisms Earth had ever hosted. They did not roar, charge, or soar. They built nothing with bones or shells. Yet they re-engineered the planet's chemistry using nothing more than sunlight and persistence. Cyanobacteria were far from the first life forms. By the time they came to dominate, life had been around for over 1 billion years. But cyanobacteria were different in a crucial way. They had cracked the problem of oxygenic photosynthesis. This biochemical method allowed them to take in carbon dioxide and water, harness the energy of sunlight, and turn those raw ingredients into sugars. In the process, they split water molecules and vented oxygen as waste. No one instructed them to do this. They had no conscious goals. Their innovation grew out of mutations, blind trial and error, and chemical opportunism. Yet it worked, and once it worked, it spread. Oxygenic photosynthesis was far more efficient than earlier. Anoxygenic versions that relied on sulfur or hydrogen as electron donors. That efficiency gave cyanobacteria a decisive advantage. They could reproduce faster, colonize new environments more aggressively, and outcompete their neighbors. Gradually, their numbers swelled. Cyanobacterial mats developed in tidal flats, encrusted submerged rocks, and ringed the edges of shallow lagoons. Some populations drifted freely, forming a loose planktonic layer in sunlit waters. These mats were not random gatherings. They were tiered systems, frequently hosting other bacteria just beneath the cyanobacteria organisms that feasted on cyanobacterial waste products or exploited different wavelengths of light. Together, these layered communities formed robust, highly organized ecosystems. They created gradients in light, oxygen, and nutrients. They cycled elements through tightly integrated networks. Over time, they also contributed to the formation of stromatolites, those domed, banded structures that still appear in ancient sedimentary rocks. Stromatolites were not mere geological oddities. They were the preserved footprints of microbial activity built up over thousands of years. They accumulated layer by layer as mats trapped grains of sediment, secreted minerals, and shielded themselves with slimy protective coatings. The result was a laminated column, blending biology and geology, a slow, stubborn monument to endurance. In the early Proterozoic, stromatolites were common. They dotted shallow seas, extended along coastlines, and lined reef-like edges of platforms. They represented one of the planet's main ecosystem types, not towering, but widespread. These structures include some of the earliest macroscopic life forms visible without magnification, and many were created by cyanobacteria or organisms much like them, but their deeper legacy lay in the atmosphere. The oxygen these microbes exhaled didn't immediately transform the air. For a long time, it was absorbed by dissolved iron, volcano-fed gases, and decaying biomass. For millions of years, the atmosphere's overall makeup remained largely the same. As the iron rusted out and the other chemical sponges became saturated, though, that changed. Iron concentrations in the oceans fell. Reactive volcanic gases diminished or were consumed. Eventually, oxygen had nowhere else to go but upward into the sky. Cyanobacteria did not pause. They continued exuding oxygen, oblivious to the long-term revolution they were driving. They weren't trying to rework the world. They were simply following their metabolic programming. But they became the agents of one of Earth's defining transformations. The oxygen they generated began to accumulate aloft, gradually rearranging the atmosphere and triggering new environmental feedback loops. One of the earliest planetary-scale impacts hit the climate. Methane, then a major greenhouse gas, is extremely sensitive to oxygen. As oxygen levels increased, methane broke down more readily. Less methane meant a weaker greenhouse blanket, which contributed to global cooling. It's likely that Earth's first large-scale ice ages were linked, at least in part, to the success of cyanobacteria and the atmospheric changes they drove. Their effect on life was even more dramatic. For many strictly anaerobic microbes, oxygen was a deadly threat. It interfered with core biochemical machinery, derailed energy production, and damaged genetic material. Entire microbial ecosystems were thrown into upheaval. Communities collapsed, retreated to deeper, more secluded environments, or shifted into zones where oxygen couldn't easily reach. Others adapted. The so-called oxygen crisis forced evolution onto new tracks. Some lineages developed antioxidant defenses. Others took a more radical path. They learned to use oxygen. Aerobic respiration emerged, providing a much more efficient means of extracting energy from food. That extra energy budget opened the door to faster metabolisms, more elaborate cell structures, and ultimately, more complex organisms. Cyanobacteria were not an evolutionary endpoint. They were a spark. They forged conditions that allowed eukaryotic cells to appear, and, eventually, multicellular life to blossom. Their presence transformed the biosphere, reshaped the oceans, and rewrote the atmosphere. They never knew they were authoring a new chapter in life's story. They just were. Modern descendants of those organisms still surround us. You'll find them in ponds, spread across whetstones, thriving in hot springs, persisting even in deserts. They are the green film on stagnant water, the slippery coat on a river rock. They lack glamour, but they are ancient, tough, and still running essentially the same biochemical playbook that altered the planet eons ago. Every breath you draw contains oxygen that exists because of them. Not only because of what cyanobacteria are doing today, but because of what their ancestors accomplished back in the Proterozoic when they draped the planet in thin blue-green layers and quietly set in motion one of the most important planetary transformations in Earth's history. For something so central to life today, oxygen had an incredibly clumsy entrance. It wasn't invited, it wasn't anticipated. And for many of Earth's earliest inhabitants, its arrival was a serious hassle. What we now call the Great Oxygenation Event, or GOE, was not a celebration. For much of the microbial world, it was a slow-moving disaster. It started innocently. Cyanobacteria, as we've seen, had been steadily releasing oxygen into their surroundings through photosynthesis. At the beginning, that oxygen never lingered. It was immediately swallowed up by iron dissolved in seawater, by gases spewed from volcanoes, by decaying organic matter. It was like trying to fill a basin lined with thirsty sponges. Every bit of oxygen poured in was soaked up at once. For millions upon millions of years, essentially none of that oxygen made it into the atmosphere in any significant way. It vanished into the ambient chemistry of Earth's surface and oceans. But no sponge can hold water forever. Eventually, the sinks began to saturate. Iron was locked away in banded iron formations. Volcanic gas emissions changed, slowed, or became less reactive. The ocean's chemistry gradually shifted. And then one day, though, no one at the time was keeping track of, day's oxygen started to stick around. It didn't vanish on contact. It began to accumulate above the seas. This still wasn't sudden. Oxygenation almost certainly took tens of millions of years. On human timescales, that's near infinite. On geological timescales, it's abrupt. For many anaerobic organisms' life forms that had evolved in an oxygen-free world, it was catastrophic. Oxygen is a highly reactive chemical. It attacks proteins, fats, and DNA. In small amounts, it creates oxidative stress. In larger quantities, it becomes toxic. For microbes adapted to strongly reducing environments, the spread of oxygen was like flooding their habitat with a slow-working corrosive. Whole microbial communities collapsed or were forced into narrow refuges. Anaerobic bacteria retreated into deeper sediments, hydrothermal vent systems, and other oxygen-poor pockets. Some evolutionary branches may have been pruned away entirely, leaving behind nothing but faint chemical signatures in rocks. On a planetary scale, the consequences were just beginning. Methane, one of the main greenhouse gases that had been keeping Earth temperate beneath a faint young sun, began to break down in the presence of oxygen. As methane levels dropped, the planet lost some of its thermal blanket. Surface temperatures fell. Ice began to take hold at the poles, and likely crept toward lower latitudes. Earth may have experienced its first planet-wide or near-planet-wide glaciation, or at least a steep climatic downturn. This interplay between life and climate, between what microbes exhaled and how warm the planet stayed, marked a new chapter in Earth's history. Biology had begun to steer climate in a feedback loop that was dynamic, intertwined, and occasionally brutal. Oxygen also reshaped the upper atmosphere. Some of that oxygen was converted into ozone, a molecule composed of 3 oxygen atoms. Ozone is unstable near the ground, but higher up it performs a critical job, absorbing a large fraction of the sun's harmful ultraviolet radiation. Before the GOE, life could survive only underwater or deep within rocks because the surface was blasted with unfiltered UV rays. With ozone slowly building in the stratosphere, the surface started to soften. It became less immediately destructive, more forgiving for shallow water, and much later, land-based life. This set up a feedback loop. More oxygen meant more ozone. More ozone meant less UV radiation. Less UV meant safer conditions for photosynthesizers near the surface. Safer conditions allowed more biological productivity. More productivity could lead, in time, to more oxygen. The Great Oxygenation Event did not proceed in a straight line. After oxygen first climbed to meaningful levels, the atmosphere appears to have entered an unstable period. Oxygen likely rose in pulses, then fell, then rose again. Biological innovations or large geological events could have dampened or amplified oxygen levels. The mid-Proterozoic atmosphere was not a smooth, well-mixed, steady environment. For living things, flexibility and tolerance were a matter of survival. The word oxygenation sounds almost clinical, even benign. But to many of Earth's earliest life forms, this was closer to a long chemical poisoning followed by a drawn-out recovery. Even among the lineages that survived, it demanded change. New metabolic paths had to evolve. Some microbes developed enzymes that could neutralize oxygen's damaging effects. Others went further and began using oxygen as part of their energy systems, evolving aerobic respiration. This mode of respiration is far more efficient than anaerobic approaches. It allows organisms to strip far more usable energy from each molecule of food. That extra energy reserve opened the door to larger cells, more complex internal architecture, and more robust repair mechanisms. Eventually, one lineage developed internal compartments, including one dedicated to processing oxygen and generating energy, the mitochondrion. That particular innovation lies further ahead in our story. For now, oxygen was still testing the boundaries of where it could exist. It slowly climbed in the atmosphere. It seeped into soils and sediments. It induced new kinds of chemical weathering, reshaping rock surfaces and landscapes. The so-called red beds, oxidized sedimentary rocks stained with iron oxides, are a legacy of this phase. They formed when iron in terrestrial sediments encountered atmospheric oxygen and turned rusty, leaving entire rock layers tinged with red. It feels strange to frame oxygen as an environmental toxin, but in the early Proterozoic, that is exactly what it was. It drove extinctions, reorganized ecosystems, disturbed the climate, and left unmistakable marks in the geological record. It also paved the way for the modern biosphere. At the time, though, none of that would have been apparent. The planet had crossed an invisible threshold. Life was still microscopic, barely more elaborate than before. Yet its waste product had redefined the oceans, the air, and the trajectory of evolution. Without any plan, just countless cells floating in shallow seas, absorbing CO2, releasing O2, and leaving the rest of the world to deal with the consequences. The sky above an early Proterozoic Earth would not have matched ours, at least not consistently. Depending on the moment you chose, and the particular mix of gases overhead, you might have looked up from that bare rocky surface and seen a sky tinted with colors that feel closer to science fiction than familiar weather. Not the soft blue we take for granted, but something thicker, dimmer, more variable. The atmosphere was mid-transformation, and like everything else in the Proterozoic, it was changing at a glacial pace. Before oxygen truly established itself, Earth's air was dominated by gases such as methane, carbon dioxide, water vapor, nitrogen, and hydrogen, with a hefty dose of sulfur compounds from volcanic eruptions. Ultraviolet radiation poured in nearly unimpeded. Without a substantial ozone layer, there was nothing to block it. And without abundant oxygen, there was no straightforward path for ozone to form in the first place. Methane in particular played a starring role. Besides its greenhouse properties, methane has interesting optical effects. In high concentrations, it can spawn a photochemical haze, a thick soup of organic particles called tholins. On the early Earth, it's possible that such a haze draped the atmosphere, scattering sunlight and lending the sky orange or brown tones. Under such conditions, the world might have resembled a continuous late afternoon glow or a smoky twilight, depending on haze density and the sun's angle. Then oxygen began to build. As free oxygen accumulated, it reacted with methane, breaking it down. Methane levels dropped, and with them the persistent atmospheric haze began to thin. As the haze dissipated, the scattering of light changed. The atmosphere started behaving more like the one we know today, allowing the shorter blue wavelengths of sunlight to dominate the daytime sky. Pinpointing exactly when the sky became consistently blue is difficult. The transition was not a simple on-off switch. Instead, it was a gradual eroding of the orange-brown haze and a slow amplification of clear, Rayleigh scattered blue. Sunlight passed through an evolving mixture of gases. The blue we associate with a well-oxygenated sky emerged over extended time as oxygen concentrations rose, methane fell, and significant ozone formed in the higher layers of the atmosphere. Ozone is born when ultraviolet light splits oxygen molecules, O2, into single oxygen atoms, which then recombine with other O2 molecules to produce ozone, O3. Once this cycle was underway, a new protective blanket formed high overhead. For the first time, Earth had a dedicated filter that could intercept much of the UV radiation which had previously hammered the surface. With that shield, shallow water and eventually exposed land became marginally less hostile. Not truly safe, not yet, but less lethal. Organic molecules survived longer without being shattered by UV. Mutations still occurred, of course, evolution never paused, but the environment was less instantly destructive. This shift in turn made it easier for microbes to occupy near-surface and sunlit environments. Visually, these atmospheric changes would have been striking if any eyes had been around to notice. Over time, the sky would have cleared, shadows would have sharpened, color contrasts would have grown more vivid. Sunsets might have begun to show the layered reds and oranges we recognize today. But there were no animals to squint at the brightness, no viewers to be moved by the beauty. Just microbial communities quietly performing chemical routines while, above them, the character of the sky evolved. At the same time, the temperature balance was changing. With methane decreasing and carbon dioxide taking up more of the greenhouse burden, the atmospheric thermostat shifted. Because methane is a stronger heat trap than CO2, its decline helped cool the planet. Over the longer term, this cooling trend likely contributed to the severe ice ages that wrapped much of the planet in snow and ice, possibly from pole to equator. Before the glaciers spread, though, the light changed first. Rocks cannot remember sky color directly. No sediment holds a snapshot of what the heavens looked like over the first continents, or how daylight glimmered on early stromatolite reefs, but they provide hints. Those red bed sediments stained with oxidized iron tell us oxygen had become a substantial part of the air, enough to rust soils, enough to oxidize minerals, enough to imprint a permanent hue on the planet's surface geology. It's odd to realize that the sky's color is a function of chemistry. We assume blue is normal, sunsets are red, and night is black, punctuated by stars. Yet that entire visual palette depends on atmospheric composition, on the scattering and absorption of light by molecules overhead. In the Proterozoic, the sky was still in flux. Slowly, it settled into the appearance we now see as normal. Below that changing dome, life went on. Microbes were largely indifferent to the visual drama, but they benefited from the chemical and radiative changes all the same. Oxygen still hadn't finished its ascent. It continued to rise, and with it came new pressures, new forms of adaptation, and eventually, an entirely new type of cell. They had no architectural plans, no written instructions, no tools, no conscious design. Yet these ancient microbial mats were the first communities to etch enduring structures into the landscape. Long before coral reefs, termite mounds, or human-built cities, layered bacterial populations were constructing something surprisingly elaborate. Cooperative living platforms, built one cell at a time, spread across shallow seafloors, tidal flats, lagoon margins, and continental shelves. Anywhere the water was calm enough and sunlight could reach, these mats took hold. Initially, they were thin, just films of photosynthetic microbes stuck to rock or sediment glued together by slimy secretions. But over long periods, they became more complex. They stacked upward. They developed vertical zoning layers sorted by light levels, oxygen availability, nutrient gradients, and waste products. It was a type of zoning enforced by chemistry, not laws. The topmost layers were dominated by cyanobacteria. They bore the main burden of capturing sunlight, splitting water, producing oxygen, and manufacturing organic compounds. Beneath them lived other microbial specialists, anaerobes, that consumed the waste products of the photosynthesizers, sulfur eaters, nitrogen fixers, fermenters. Every microlayer had a role. Deeper still, where oxygen dwindled or disappeared, metabolism shifted again. Instead of generating oxygen, These organisms decomposed material, recycled nutrients, and closed the loop. It wasn't a strict hierarchy so much as a cycle production at the top, consumption and recycling below. Some mats trapped grains of sand and silt that currents or tides swept across them. Others precipitated minerals directly from seawater as byproducts of their metabolism. Over time, these processes gradually built upward, forming laminated, often dome-shaped or columnar structures, stromatolites. Some formed isolated knobs, others stretched for kilometers, carpeting broad coastal platforms in patchy microbial quilts. Stromatolites still exist today in a few special environments: Shark Bay in Australia, certain hypersaline lagoons, some alkaline lakes. Modern examples grow very slowly, usually in harsh conditions where grazers and seaweeds are scarce. They are lingering echoes of a time when such structures were widespread and largely unchallenged. In the Proterozoic, stromatolites were among the most prominent features of shallow marine landscapes, not because they towered high, but because almost nothing else built long-lasting mineralized frameworks on that scale. They were living, geochemical machines. Each layer performed particular tasks, and the whole structure regulated its own internal chemistry. Oxygen was efficiently made and consumed. Sulfide was generated and then used. Carbon cycled continually through photosynthesis, respiration, and fermentation. From the outside, a mat might look like little more than slimy film or lumpy rock, but internally it was dense with activity. These mats affected their surroundings in profound ways. They helped stabilize sediments, trapping particles and shaping microhabitats. They changed the composition of nearby water, altering acidity and redox conditions. They were, in a sense, ancient city systems with specialized zones, internal circulation, waste handling, energy generation, and defense strategies. Even without true predators yet, competition was present. Different bacterial strains jostled for space, light, and nutrients. Some mats produced chemical antibiotics to deter invaders. Others colonized deeper or slightly dimmer waters to avoid direct conflict. Evolution didn't wait for big-brained animals to start getting inventive. The architecture of these mats wasn't made of bone or stone, but it was durable enough to fossilize. Stromatolites preserve especially well in carbonate-rich environments. Their layers eventually turn to rock. Some hold microscopic fossils, tiny threads, spheres, or filaments hinting at the organisms that built them. Others hold only textural patterns. In both cases, they offer more than just a presence-absence record. They reveal clues about ancient oxygen levels, light penetration, and nutrient gradients. They help identify which microbes were successful and what kinds of environments supported complex communities. They also mark time. Each layer accreted slowly, so counting, measuring, and analyzing stromatolitic laminations gives geologists a way to reconstruct environmental histories spanning millions of years. There's something quietly astonishing about how long these mats dominated. For much of the Proterozoic, microbial mats were the planet's primary ecological framework. Not a side note, not a fringe habitat, but the main event, sustained for over 1 billion years. Their slow, coordinated, layered strategy was so effective that there was little pressure to change, so it persisted. Eventually, new cell types and grazers would emerge. They would nibble away at the mat's stability, physically disrupting them. But that was still far in the future. For now, in the mid-Proterozoic, microbial mats and stromatolites were the chief ecosystem engineers, sculpting coastlines, setting chemical conditions, and building quiet cities of slime, one layer at a time. For most of Earth's story so far, life had no eyes, no mouths, no spines, no bones, no wings slicing through air or fins propelling through water. There was motion, but not in the sense of animals darting or fleeing. Nothing stalked, nothing ran. There was no sensory awareness beyond molecules and membranes reacting to gradients. And yet, life flourished. The Proterozoic was ruled by microbes. Everything was small. Nearly everything existed as a single cell. Diversity was huge, but it played out entirely at microscopic scales. Not because larger body plans were physically impossible, but because nothing had yet pushed evolution hard enough in that direction. There were no predators to chase, no prey to escape, no arms races requiring speed, coordination, or centralized control. Microbes dominated through chemistry rather than behavior. They altered their surroundings by existing, breathing, excreting, exchanging molecules. Each cell functioned as a tiny chemical plant, running ancient scripts. Take in, transform, release. There were no faces to read or limbs to gesture with, but there was communication. Cells sensed and responded via chemical signals. Gradients of nutrients or toxins told microbes where to swim, when to divide, when to form clusters. Some cells possessed flagella whip-like appendages that allowed them to paddle toward food sources or away from unfavorable conditions. Others floated passively on currents or clung tenaciously to substrates. Life didn't need to be fast. It needed to be efficient. Lacking eyes doesn't mean lacking light sensitivity. Many microbes reacted to light, especially those that photosynthesized. Cyanobacteria adjusted their vertical position in the water column to capture optimal wavelengths. Some likely shifted depth in response to daily light changes or fluctuations in water clarity. They couldn't see in any conscious sense, but they had photoreceptors molecular systems that altered cell behavior when struck by photons. The microbial biosphere was shaped by gradients. At the top, light, oxygen, and relatively high energy. Below, darkness, anoxia, and slower metabolisms. Temperature, acidity, salt levels, all varied spatially, creating layers and patches. Microbes specialized to occupy narrow bands where particular conditions overlapped. Some thrived in extremes: scorching vents, salty brines, highly acidic pools, crushing pressures. The variety was immense, despite the absence of organs or body plans. This microscopic world was far from static. Genetic change was constant. Bacteria reproduce quickly, and every division carries a chance of mutation. Multiplied over millions of years, that leads to extraordinary diversification. Horizontal gene transfer, where microbes pass genes directly to one another, accelerated this diversification further. Traits like photosynthesis, nitrogen fixation, or toxin resistance could spread laterally across lineages, not just down family trees. Evolution didn't bide its time waiting for larger, more complex life. It took every shortcut available. The fossil record from this interval is sparse because soft microbial bodies decay quickly and rarely mineralize, but they left behind chemical clues, biosignatures. Certain complex organic molecules, such as specific hopanoids or steranes, point to the presence of particular groups of bacteria or early eukaryotes. Isotope ratios in carbon, sulfur, and nitrogen hint at which metabolisms were operating. Who was fixing carbon from CO2? Who was oxidizing sulfur? Who was cycling nitrogen? Sometimes sediments preserve microscopic textures, filaments, lenticular patches, or layered fabrics that represent ancient microbial mats or communities. They are not photographs, but they're enough to reconstruct a world teeming with unseen life. Ecologically, the system was self-contained. Photosynthetic primary producers captured energy and built organic molecules. Other microbes consumed those molecules. Decomposers broke down the remnants and recycled nutrients. It was a complete food web, just stripped of visible spectacle. No jaws, no dramatic hunts, just molecules flowing through networks of cells in mud and water continuously. Time passed differently at this scale. Without nervous systems, there was no awareness of before or after, no personal memory. Cells reacted directly to local conditions, a new gradient, a signal, a chemical change, then a response. That was life's tempo. For all its simplicity, it was remarkably robust. This microbial regime lasted for more than 1 billion years. There were no global collapses of the sort that would later devastate animal life. There were shifts, yes, oxygen levels changed, climates swung, oceans froze, and thawed. But the microbial biosphere adjusted, closing loops, occupying new niches, recycling everything. It's tempting to treat this period as a warm-up, a long preface before real life arrived. But evolution has no main act. There is no final destination. The microbial world was complete on its own terms, intricate, fine-tuned, and overwhelmingly successful. Many of the lineages that dominated then are still with us. Some have changed very little. Their descendants live in your intestines, in your garden soil, in the open ocean. Others eventually gave rise to entirely new kinds of organisms, eukaryotes, multicellular bodies, animals. But in the stretch of time we're inhabiting now, those futures hadn't materialized yet. This was the microbes era, a planet governed by beings too small to see, building chemical empires, thriving without senses, without consciousness as we understand it. And yet, they were the most powerful biological force on Earth. Their reign wasn't just long, it was foundational. And somewhere within this mostly invisible world, a new kind of cell began to emerge, one that carried a nucleus. They didn't burst onto the scene with fanfare. No clean split, no momentous boundary, no ancient flag thrust into the mud to mark their arrival. Eukaryotes, the kind of cells that would eventually build every plant, animal, fungus, and all the complex life in between, appeared quietly. Like so much in the Proterozoic, their origin wasn't abrupt. It was slow, convoluted, and wrapped in uncertainty. But when they did arise, they represented a radical departure from everything that had existed up to that point. A eukaryotic cell is more than a blob of cytoplasm inside a membrane. It is internally partitioned, organized, and specialized. It possesses a nucleus, a protected compartment wrapped in its own membrane, where DNA is stored and managed. It contains organelles, miniature internal structures dedicated to particular tasks, mitochondria for energy production, endoplasmic reticulum for processing proteins and lipids, Golgi bodies for packaging and transport, a cytoskeleton to maintain shape and coordinate internal movements. This wasn't just a bag of reacting chemicals anymore. It was a system with traffic routes, storage hubs, quality control, and regulation. None of these features existed in earlier life. Prokaryotes' bacteria and archaea are spare by comparison, incredibly efficient but minimal. Their DNA is not sequestered in a nucleus. Many reactions happen wherever the right substances bump into each other. That simplicity has worked astonishingly well for billions of years and still does. But eukaryotes took a different path. They invested in internal complexity. How did that happen? The leading idea centers on symbiosis, a merger between distinct cells. At some point, an archaeal host cell took in a bacterial partner. It didn't digest it. It kept it. Maybe the host engulfed the bacterium as potential food but failed to finish the job. Maybe the bacterium slipped inside through a crack or intimate association and began offering useful metabolic services. Perhaps it excelled at using oxygen to generate energy. Whatever the exact mechanism, the end result was coexistence. The bacterium stayed. Over time, it surrendered many of its genes to the host's nuclear genome. It lost its independence and the ability to live alone. In return, it became an organelle, the mitochondrion. The host cell gained a powerful internal energy plant. This union wasn't sudden or clean. It unfolded over immense spans of time. But once established, it changed the rules. Mitochondria provided a consistent, efficient source of ATP adenosine triphosphate, the universal energy currency of cells. In return, the host supplied raw materials, shelter, and control over reproduction. Mitochondria still bear the traces of their bacterial ancestry. They contain their own small circular DNA, separate from the nuclear chromosomes. They divide independently inside the cell in a way reminiscent of bacterial fission. They have a double membrane, one from their original bacterial envelope, and another likely inherited from the host's engulfing process. Their ribosomes and some of their proteins look more bacterial than eukaryotic. This deep merger marked a pivotal step, not because it made cells fancier, but because it made them energetically richer. Aerobic respiration, channeled through mitochondria, yields far more ATP from each molecule of food than anaerobic pathways. That surplus budget allows cells to support larger genomes, maintain elaborate internal infrastructure, run complex regulation, and perform multiple tasks simultaneously. Complexity became not just possible, but sustainable. The first eukaryotes were still single-celled. They didn't yet build bodies or tissues. They had no limbs, no nervous systems, no obvious external features. But they had the potential for all of that. They began to diversify. Some lineages became ancestors of algae. Others drifted toward what would one day become fungal groups or animal lines. Many experimental branches arose only to disappear without descendants. But a few persisted and radiated, filling out the early eukaryotic tree. This era is marked not by eukaryotes dominating ecosystems, but by innovation. Bacteria and archaea still ruled numerically and metabolically. Eukaryotes were minority newcomers. But they were trying different things. Some developed flexible cell membranes and the ability to engulf other cells entirely. This opened the door to true predation. For the first time, life was not only absorbing dissolved nutrients. Some cells were actively consuming other cells. That new relationship predator and prey created fresh selective pressures. Pressure to detect, to escape, to pursue, to defend. Eukaryotes didn't evolve eyes yet, but they needed expanded sensory abilities, more subtle chemical detection, more nuanced responses, greater mobility. Life began to stretch into space as a concept, not just chemistry. Eukaryotes also built more complex partnerships. Some became hosts to additional symbionts. In lineages that would lead to algae and plants, another critical merger took place. A photosynthetic cyanobacterium was engulfed and retained, separate from the mitochondrial event. Over time, this symbiont turned into a chloroplast, the organelle responsible for capturing light and powering photosynthesis in modern plant cells. Once again, the storyline was similar. Collaboration, integration, then specialization. Chloroplasts, like mitochondria, keep their own DNA and double membranes, clear markers of their bacterial roots. Symbiosis, once brushed off as a biological curiosity, is now seen as a powerful evolutionary engine. It's not just about survival, it's a creative process. Instead of excluding outsiders, early eukaryotes repeatedly incorporated them, turning former free livers into essential internal machinery. In doing so, they redefined what a cell could be. The mitochondrial origin story complicates the old picture of evolution as pure competition. Yes, natural selection eliminates the poorly adapted. But some of the most transformative steps in life's history came from cooperation so deep it erased the boundary between partners. The fittest weren't always those that fought best, but those that could knit themselves together with others. Even today, mitochondria sit at the center of eukaryotic life. They power muscle contraction, brain activity, development, reproduction. Almost every system you can name depends on them. When mitochondrial function falters, entire organisms suffer. When it thrives, life can support movement, thought, and complexity. Despite their integration, mitochondria remain slightly apart. Each one carries its own genetic fragment, a microscopic fossil of a time when it was a free bacterium in an ancient sea. They have no awareness of that past, no memory, no intent. But the lineage is unbroken. Every mitochondrion in every animal, plant, or fungus can trace its ancestry back to that original bacterial endosymbiont captured more than 1 billion years ago by a cell that did not destroy its meal but kept it. In the Proterozoic, that moment left no dramatic monuments or visible scars. It didn't stir the oceans or move the continents, but it initiated the age of complexity, cells becoming composite systems, life deepening inward before it ever grew outward. At some point in the later Proterozoic, Earth veered toward a profound chill. The interior kept churning, volcanoes still erupted, plates continued to creep over the mantle. But from the standpoint of climate, the planet slipped dangerously close to shutting down. For reasons we still don't completely understand, Earth tipped into an intense freeze. Ice spread outward from the poles, pushing into lower latitudes. Temperatures plunged. Sea ice thickened. possibly sealing over vast stretches of ocean beneath rigid, white armor. For a while, the planet may have looked less like a vibrant blue world and more like a frozen satellite glittering bright and nearly locked in cold. This was more than just a cold spell. At least twice during the Proterozoic, and perhaps more often, the planet experienced glaciation so extreme that they covered much of the globe. Not just polar caps and alpine zones, but areas near the equator, shallow tropical shelves, and broad continental interiors. The label we use for this scenario is Snowball Earth. Although scientists still debate how complete the ice coverage was, the evidence strongly indicates something extraordinary. The proof lies in rocks. Glacial deposits of this age appear in locations that, based on paleomagnetic data, sat near the equator at the time. Drop stones, outsized clasts carried by ice and then released into fine marine sediments as icebergs melted, are found embedded in ancient seafloor layers far from what would be modern polar regions. Scratches and grooves carved into bedrock by moving glaciers, glacial striations, are seen in terrains that are now warm and temperate. All of these geological signs converge on a world where ice extended into regions that today are considered tropical. So what set off such a severe freeze? Climate is the result of a delicate balance among solar energy, atmospheric composition, ocean circulation, and planetary reflectivity. During the Proterozoic, the sun was still dimmer than it is now, emitting roughly 94% of its present energy. That alone made the climate more vulnerable to cooling. Then consider the atmosphere. Earlier in the Proterozoic, methane played a crucial role as a greenhouse gas, supplementing carbon dioxide to keep the surface warm. But as oxygen levels rose due to cyanobacterial activity and other photosynthesizers, methane was oxidized and removed more rapidly from the air. CO2 was still present, but if enough of it was drawn down by chemical weathering of rocks or biological uptake, greenhouse warming could weaken. Once ice began to form, feedback took over. Ice and snow reflect more sunlight than dark ocean or bare rock. The more of Earth that turned white, the more sunlight bounced back into space, and the less heat was absorbed. That meant more cooling, more ice, and so on. If this positive feedback passed a certain point, it could lock the planet into a deep freeze. The expansion of ice likely affected the carbon cycle too. With much of the surface frozen, weathering rates would have dropped. Less weathering means less CO2 removal from the atmosphere, something that would eventually matter for recovery, but initially the overwhelming reflectivity may have outpaced that balancing factor. The result? Near global or global glaciation. What happened to life during these frozen intervals is harder to reconstruct. Fossils from this time are scarce, and the biosphere was still mostly microbial. Shallow marine environments. The home turf of photosynthetic mats and early eukaryotes must have been severely stressed. Thick ice would have cut off most sunlight, throttling the energy supply to surface ecosystems. Still, life did not vanish. It adapted and retreated. Beneath the sealed oceans, the deep waters remained liquid. Heat from Earth's interior continued to warm the bottom. Hydrothermal vents on the seafloor still spewed mineral-rich fluids. Around these vents, chemotrophic communities, organisms powered by chemical energy, not light likely, continued with little interruption. They used the oxidation of substances like hydrogen sulfide or methane to drive metabolism, indifferent to the global winter above. In shallower regions, conditions were more marginal but not uniformly lethal. Some models suggest that even during maximum glaciation, thin zones of open water, or at least thinner ice, persisted in places called polynyas. Wind, ocean currents, or geothermal currents could have prevented complete freezing in localized pockets. In those small refugees, photosynthetic microbes might have managed to cling on, catching what little light filtered through. Even where the ice was continuous, it may not have been a solid uniform slab. Beneath thick sea ice, brine channels and slushy layers can form, creating dim, cold, but habitable microenvironments. Modern analogs beneath Antarctic ice shelves support microbial communities that photosynthesize under faint light or rely on chemical energy instead. It's reasonable to think something similar existed beneath the Proterozoic snowball. On land, visible life was almost entirely absent even before the freeze. But microscopic life may have survived within rocks endoliths tucked into tiny cracks, protected from harsh radiation and extreme surface cold. Today, microbial communities inhabit such niches in the driest Antarctic deserts and deep underground. They live at a crawl, metabolizing slowly, dividing only occasionally, but they endure. Other organisms may have adopted forms of dormancy. Spores, cysts, and resistant resting stages can wait out unfavorable conditions for remarkably long periods. Some early eukaryotes likely used such strategies, shutting down most metabolic activity, preserving internal structures, and reawakening only when temperature and light allowed. In catastrophes like this, evolution tends to favor not the most aggressive or complex, but the smallest, the toughest, and the most patient. Cells capable of going into low-power mode slowing metabolism, tolerating nutrient scarcity, and withstanding environmental swings stood the best chance of surviving the frozen ages. Eventually, the freeze began to loosen. Volcanic activity, unaffected by surface ice, continued beneath the crust. Volcanoes vented CO2 into the atmosphere, but with much of the surface buried under the ice, there were fewer ways for that CO2 to be drawn back down by weathering or biology. Over millions of years, the gas accumulated, building up the greenhouse effect. At some threshold, rising CO2 tipped the balance back. Temperatures crept upward. Ice began to melt at the edges, exposing darker water and rock, which absorbed more heat and accelerated warming. Meltwater surged into the oceans. The transition from a snowball climate to a warmer world was likely violent. Rapid warming, intense storms, heavy rainfall loaded with carbonic acid from high CO2 air, widespread erosion, and dramatic sea level changes. Nutrients, flushed from freshly exposed rock and deglaciated soils, poured into the oceans. This influx of phosphorus and other key elements may have triggered enormous blooms of photosynthetic life. Oxygen production ramped back up. Ecosystems that had been compressed into refugees expanded, exploring habitats that had been reset by ice. The end of a snowball. Earth wasn't just a return to business as usual. It was a planetary upheaval followed by opportunity. Chemical weathering resumed with vigor. Oxygen levels likely rose further. Ocean circulation patterns changed. Communities reorganized. In the wake of this environmental chaos, new evolutionary possibilities opened. These global glaciations are often cast as disasters, and in many respects they were. Massive stress, probable die-offs, and deep disruption of global cycles. Yet they also acted as resets. They may have cleared out sluggish ecosystems, opened ecological space, and encouraged innovations. Not because ice is inherently good for life, but because life is resourceful enough to adapt around it and then exploit the new world that follows. The Proterozoic was not a peaceful plateau. It included its own bouts of chaos, global rusting of the oceans, atmospheric revolutions, and now planet-encompassing freezes. Each time, life persisted, sometimes barely, but it persisted. The cold narrowed life's options. It forced organisms into remote corners and low-energy strategies, but it did not erase them. Beneath thick, gleaming shells of ice, life waited, held on, and prepared, unknowingly, for the next round of change. Somewhere in the later Proterozoic, Earth brushed close to a kind of environmental standstill. Not in its interior. The core still convected, volcanoes still erupted, plates still drifted. But at the surface, the climate system was pushed into some of the harshest conditions the planet has ever seen. Ice advanced, temperatures plunged, the oceans sealed under frozen lids. For a stretch of time, Earth could almost have passed for a frozen moon. The snowball Earth phases did not wipe life away. They tightened the noose, restricted possibilities, and demanded new strategies. The biosphere responded less with defiance than with patience. During these deep freezes, the planet's skin looked almost alien. Continents lay hidden under enormous ice sheets. Oceans were topped by ice that may have reached hundreds of meters in thickness. The air was thin and dry, whipped by strong winds. With the sun's already reduced output and a shining global mirror of ice, solar energy was mostly reflected back into space. Photosynthesis. The main energy engine at the surface was throttled almost to nothing. What little light reached Earth either bounced off snow and ice or was dimly filtered through them. Yet the entire world wasn't inert. The ocean beneath the ice remained liquid and in motion. Heat leaking from Earth's interior continued to warm the deep waters. Hydrothermal vents on the seafloor, as always, gushed hot, mineral-laden fluids, providing small but stable oases of chemical energy. In these dark depths, microbial ecosystems hardly noticed the ice age overhead. Vent communities already relied on chemosynthesis rather than sunlight. Bacteria and archaea oxidized hydrogen sulfide, methane, and other compounds to drive their metabolisms. For them, the snowball above was another distant variable. Closer to the frozen surface, life walked a finer line. If thin regions of ice or occasional open water patches persisted, maintained by winds, currents, or geothermal hotspots, then photosynthetic microbes could still operate there, in narrow belts of weak sunlight. Beneath the ice, slushy layers and brine channels may have offered dim environments where microscopic communities clung to the underside of the ice, or hovered in salty pockets. Similar habitats exist today below Antarctic sea ice, and they support thriving microbial life at near-freezing temperatures. On the continents, macroscopic life was absent, but microbes almost certainly persisted out of sight. Within rocks, in fractures, and deep under frozen soils, endolithic communities could survive on tiny dribbles of energy. They metabolized slowly, dividing rarely, but they endured. Some microbes can enter nearly suspended states forming spores or resting cysts to get through extended stress. Early eukaryotes may have relied on such dormancy to ride out the worst of the freeze, waiting for conditions to improve. Through all of this, the winners weren't the largest or most sophisticated, but the smallest and most economical. Organisms that could lower their energy demands, tolerate long periods of scarcity, and withstand environmental extremes stood the best chance of persisting. When warming finally arrived, it came with drama. Volcanic CO2 had been accumulating for millions of years while the ice suppressed weathering and biological uptake. At some point, greenhouse warming overpowered the cooling from ice reflectivity. The climate tipped back. Ice began to melt along coasts and cracks first, then more broadly. Exposed dark surfaces soaked up more sunlight, further accelerating the thaw. The deglaciation was almost certainly rapid on geological timescales. Gigantic floods cascaded off shrinking ice sheets. Newly exposed rock surfaces began weathering vigorously, reacting with CO2 in the atmosphere. Rain, likely acidic from high CO2 levels, beat down on bare ground. Rivers surged with sediment and dissolved minerals, flushing vast quantities of nutrients into the oceans. For marine life, this was a windfall. Nutrients like phosphorus and iron, previously locked under ice or in weathering-resistant surfaces, mixed into sunlit waters. Microbial photosynthesizers exploded in number. Oxygen production spiked. The biosphere, which had been compressed into tiny refuges, expanded into a reset seascape. The aftermath of snowball Earth events was thus a mix of chaos and opportunity. Rapid warming, intense erosion, chemical shocks, and also ecological renewal. Life that had waited at the margins now spread into newly viable niches. Oxygen levels ratcheted up again. Ocean chemistry shifted toward conditions more favorable to complex eukaryotes. Some of the earliest convincing fossils of multicellular eukaryotes appear in rock layers deposited soon after these great glaciations. They are not animals, at least not in the modern sense, but they show organized multicellularity, multiple cell types, size beyond typical single cells, and evidence of coordinated growth. It's not that ice directly manufactured complexity, but that the environmental reset allowed those already evolving lineages to spread and diversify. Survival through these deep freezes wasn't about conquering the cold. It was about enduring it, shrinking, slowing, hiding. When the ice retreated, survivors emerged into a world chemically and physically transformed. Their experiences had filtered the biosphere, leaving behind lineages that had proved resilient. Those were the ones that would shape what came next. While life in the Proterozoic was grappling with frozen oceans and shifting atmospheres, the solid earth beneath it was anything but static. The continents, bare and silent to the naked eye, were on the move. Not fast enough for any creature to notice day to day, but steadily enough to redesign the planet over tens and hundreds of millions of years. Deep below the microbial mats and ice sheets, rigid plates of lithosphere crept across the globe, driven by the convective churn of the mantle. This motion wasn't random. It followed patterns spreading centers, subduction zones, collisions that produced a slow, planetary-scale choreography. Tectonic activity didn't originate in the Proterozoic. Some form of plate tectonics was likely active as far back as the Archean. But by Proterozoic time, the system had matured. Continents had thickened and become more stable. Cratons, the ancient, buoyant cores of continents, had formed and toughened. These sturdy blocks resisted deformation and erosion better than younger crust. Over eons, they crashed together, rifted apart, then collided again, gradually assembling larger continental masses. This was the age of recurring supercontinents. The concept is simple. Intervals, when most of Earth's land was clumped into one or a few large masses. Reality, however, is messy. Supercontinents don't appear fully formed overnight. They assemble piece by piece through a series of collisions, then disintegrate unevenly through rifting episodes. During the Proterozoic, several supercontinents likely formed and broke apart. One early candidate is Columbia, also known as Nuna in some reconstructions. It seems to have coalesced around 1.82 billion years ago, stitched together from older cratons by subduction zones and mountain-building events. The exact outline is still under debate, but rocks and magnetization patterns suggest that what we now call North America, Baltica, and chunks of Siberia were once joined as parts of this mega-continent. Columbia did not last indefinitely. Supercontinents trap heat beneath their thick crusts, reducing the efficiency with which Earth's interior can cool. Over time, that trapped heat contributes to rifting, the land stretches and thins, volcanism increases, and eventually the supercontinent fragments, birthing new ocean basins. After Columbia came another major assembly, Rodinia. It formed around 1.1 billion years ago with a different arrangement of familiar pieces. North America sat near its center. Around it clustered components we now recognize as parts of South America, Africa, Australia, and Antarctica. Though their exact configuration remains an active research area. Rodinia's formation coincided with huge orogenic events, mountain-building episodes such as the Grenville orogeny. These collisions built high ranges, thickened crust, and produced belts of metamorphic rocks that still outcrop today. As these mountains rose, they began eroding, sending rivers of sediment into adjacent basins. Those sediments became archives, recording environmental and biological stories in their layers. A supercontinent reshapes more than maps. It alters ocean currents, atmospheric circulation, and climate. One vast landmass disrupts wind patterns, redistributes rainfall, and modifies how heat is transported. Once Rodinia was fully assembled, it may have played a role in fostering global cooling and ultimately the snowball Earth episodes. Its later breakup, in contrast, likely injected CO2 into the atmosphere through increased volcanism and exposed fresh rock to weathering, affecting climate in the opposite direction. Tectonics also controls the recycling of materials. At subduction zones, old oceanic crust plunges into the mantle and partly melts, feeding volcanoes that spew new rock and gases. Mid-ocean ridges create fresh seafloor. Continents ride along, buckling into mountains, then wearing down again. These cycles influence the supply of crucial nutrients like phosphorus and iron to the oceans. Newly uplifted mountains weather rapidly, sending nutrient-rich sediments downstream. Those nutrients can stimulate biological productivity, potentially affecting oxygen levels and carbon burial. The links between tectonics and life run deep. Mountain ranges modify weather and rainfall, shifting erosion patterns. Erosion controls the flow of minerals and elements into the sea. Those elements feed microbes, which in turn modify atmospheric gases. Volcanism releases CO2 and sulfur gases, These alter climate and atmospheric chemistry, feeding back into evolutionary pressures. Viewed from afar, Proterozoic Earth might have seemed quiet, no forests, no animal herds, no dramatic coastlines dense with visible life. But under the surface, the engine was humming. Continents collided and cracked. Oceans opened and closed. Mountain belts rose and then were shaved down to their roots. Over time, this ceaseless motion set the boundary conditions for climate, weathered the raw materials that life would use, and shaped the habitats in which evolution unfolded. While life in the Proterozoic was dealing with global freezes and atmospheric overhauls, the air itself was evolving in ways that went far beyond simple gas percentages. It wasn't just the mixture of gases that changed, it was the tempo of the whole atmospheric system. There were no familiar daily or seasonal cycles in the sense that they dominate modern ecosystems. Instead, the atmosphere shifted in long, slow arcs that took millions or tens of millions of years to complete. Glaciations, tectonic rearrangements, biological innovations that tweaked the balance molecule by molecule. Above drifting continents and microbial mats, the air grew thinner or thicker with greenhouse gases, warmer or cooler, gradually assembling the structure that would eventually make complex life viable. After the first big rise of oxygen during the Great Oxygenation Event, things didn't settle into a clean, new equilibrium. Oxygen didn't just rise quietly to modern levels and stop. Instead, it fluctuated. It climbed, plateaued, dropped, then climbed again. It was less a smooth escalation and more a jagged climb, with long periods of relative stability punctuated by changes. Oxygen had entered the picture for good, but it remained only a small part of the atmosphere for a long time. Many estimates suggest that for much of the mid-Proterozoic, oxygen accounted for just a small fraction of today's levels, enough to oxidize iron on land and in sediments, enough to rust rocks, but not enough to support large, energy-hungry animals. This interval is sometimes nicknamed the boring billion, roughly from 1.8 to 0.8 billion years ago. It's not that nothing happened, it's that many geochemical records show long stretches without dramatic spikes or crashes. The atmosphere, oceans, and biosphere appear comparatively steady. Life was still evolving. Continents continued to move. Oxygen was present, but low. On human timescales, it might have seemed uneventful. On evolutionary scales, important groundwork was quietly underway. One critical process finding its rhythm was the carbon cycle. Carbon dioxide released by volcanoes and metamorphic processes was pulled out of the air and water by weathering and burial. Rain fell on mountainous silicate rocks, dissolved minerals, and carried them to the sea, where they reacted with dissolved CO2 and ultimately became part of marine sediments. Over long spans, this weathering draws down atmospheric CO2 and cools the planet. When weathering slows, CO2 can build up again, warming it. Tectonics fed this engine by creating mountains and fresh rock surfaces. Biology influenced it by changing the composition of soils and sediments, altering how carbon moved through the system. The result was a rough thermostat, not perfect, but capable of keeping Earth within a range where liquid water could persist. Methane, once central to keeping a faint young Earth warm, had largely ceded that role. Because oxygen reacts with methane, even modest oxygen levels curb methane's lifetime in the atmosphere. As a result, methane waned as a greenhouse driver, while CO2 became more important. That change made the climate system more sensitive to variations in tectonics and weathering. At the same time, the accumulation of oxygen enabled the formation of a persistent ozone layer in the upper atmosphere. Ozone's UV shielding effect didn't just reduce the lethality of surface sunlight, it also began to stabilize the vertical structure of the atmosphere. Layers like the troposphere and stratosphere, distinguished by their temperature profiles and chemical reactions, became more defined. That, in turn, influenced how heat and moisture circulated globally. Sulfur also entered a more intricate cycle. Volcanic emissions injected sulfur gases into the atmosphere, forming aerosols that reflected sunlight and cooled the surface temporarily. In the oceans, microbes learned to exploit various sulfur compounds, altering their oxidation states. The isotopic fingerprints of sulfur left in ancient rocks provide some of our best evidence for shifts in atmospheric oxygen, because sulfur chemistry behaves differently in low oxygen versus high oxygen worlds. Nitrogen, the dominant component of the atmosphere, gained new biological roots as well. Biological nitrogen fixation, performed by certain bacteria and archaea that can convert inert atmospheric N2 into bioavailable forms like ammonia, became widespread. This allowed life to escape nitrogen limitation in many settings, boosting productivity. More growth meant more organic carbon burial, which fed back into oxygen production and CO2 drawdown. Step by step, the atmosphere and life became entangled in a network of feedbacks. When photosynthetic organisms prospered, oxygen rose, and CO2 fell. When more organic matter was buried, less carbon returned to the air, nudging temperatures downward. When tectonic events uplifted new mountains, weathering accelerated, further adjusting CO2. Volcanoes in turn re-injected CO2 and sulfur, sometimes pushing the climate in the opposite direction. All through this slow dance, the atmosphere acquired structure. Temperature dropped with height in some layers and increased in others, governing how air masses moved. Wind patterns formed, driven by uneven heating of the planet's surface and the rotation of the Earth. Rainfall began to follow predictable large-scale routes, carving channels and basins on the continents, even without forests, grasses, or modern stormscapes. The Proterozoic air was never static. Periods of intense volcanism could cloud the skies with ash and sulfur aerosols. Intervals of continental rifting or collision could adjust CO2 levels and climate. Glacial periods pulled greenhouse gases down. Warm interludes let them rise. Late in the Proterozoic, oxygen may have seen brief surges toward values closer to modern levels, especially as multicellular life spread, though those spikes might not have held steady. Every pulse left traces: oxidized rock layers, shifts in isotope ratios, subtle changes in the types of fossils and microfossils that appear and disappear. Together, these clues tell us that the atmosphere was creeping toward a configuration that could support complex animals, even if it hadn't quite reached that point yet. Through all of this, life remained mostly microscopic. No eyes gazed skyward. No animals felt wind on skin or watched clouds gather. But the air was gradually becoming more breathable, first for microbes that could tolerate oxygen, then for cells that would rely upon it, and eventually for creatures that would sprint, swim, and fly through it. Down in the seas and sediments, cells were starting to do something new with one another, not just sharing genes or merging with symbionts, but pairing up for reproduction in a fundamentally different way. It's odd to consider, but for most of Earth's existence, reproduction was straightforward and solitary. A single cell copied its DNA, divided, and produced two nearly identical cells. That pattern binary fission was astonishingly effective. It still is. Most life today replicates this way. It's quick, reliable, and doesn't require another partner. But at some point in the Proterozoic, a more complicated strategy emerged. Not a total replacement for asexual reproduction, but an alternative. Slower, more error-prone, and strangely powerful. This was the dawn of sexual reproduction. No rock captures the first exchange of genetic material between two cells. There's no clear event we can point to as the origin of sex, yet somewhere among early eukaryotes, cells began to fuse, share chromosomes, shuffle genes, and separate again. When they did, they gave evolution a new mechanism: recombination. In a strict asexual lineage, barring mutation, offspring are clones. They carry the same genetic sequences as their parent. Evolution still happens, but it advances primarily through new mutations and occasional gene transfers. In sexual reproduction, by contrast, the genetic deck is reshuffled with each generation. Two parent cells contribute. Their chromosomes pair and exchange segments, and then are sorted into gametes. When gametes fuse, the resulting offspring bear novel combinations of traits. From an evolutionary perspective, this variation is invaluable. Sex mixes beneficial mutations into the same genome more quickly. It can help purge harmful mutations by allowing recombination and selection to separate them from beneficial genes. It also lets populations explore a broader range of genetic possibilities in a shorter time. But sex comes with costs. It requires more complex cell biology. It often demands that organisms find or recognize compatible partners. It means that each parent passes on only half of their genome to any one offspring. Errors in chromosome pairing and separation can be lethal. For sex to stick around, its long-term benefits had to outweigh these short-term drawbacks. The earliest sexual processes likely looked nothing like animal mating. They may have resembled modern microbial gene exchange. Two cells temporarily fusing their membranes, mingling genetic material, then splitting. Or one cell internalizing another, and then blending their DNA. In eukaryotes, these primitive exchanges gradually evolved into more structured cycles. Meiosis. The specialized cell division that reduces chromosome number by half likely emerged during this time. In meiosis, one diploid cell produces haploid cells, gametes, each carrying a shuffled subset of the original chromosomes. When two gametes fuse, the full set is restored, but in a new combination. This simple but powerful trick underlies the life cycles of almost all complex organisms today. Sexual reproduction probably co-evolved with many other eukaryotic traits. Cytoskeletal systems that manage chromosome movement, membranes that allow cell fusion, intricate signaling networks. Early eukaryotes may have switched between sexual and asexual modes depending on stress or resource availability, using sex to generate diversity when conditions changed. Over time, some lineages began to specialize, producing different types of gametes. Initially, these might have been similar in size, isogamy. Later, in some groups, asymmetry arose. Small, mobile gametes and large, nutrient-rich ones. The first steps toward the division we now call male and female. Evolutionary payoffs were substantial. With sex, populations could adapt faster to changing climates, shifting oxygen levels, and new ecological interactions, including predation and symbiosis. It may have helped eukaryotes weather the environmental instability of the late Proterozoic snowball Earth episodes, oxygen fluctuations, and tectonic upheavals. There was also an emergent social aspect, even if no minds were involved. Sex required cells to recognize each other, respond to signals, and coordinate timing. These forms of interaction molecular recognition, signal reception, synchronized cycles would later be critical when cells started living together not just as colonies but as integrated bodies. The physical traces of the earliest sexual eukaryotes are faint. Some large microfossils show division patterns too complex to be simple binary fission. Others resemble modern algae, many of which alternate between sexual and asexual phases. Molecular clock analyses, which compare genetic differences among living groups to estimate divergence times, generally point to the origin of sex more than 1 billion years ago, squarely in the Proterozoic. We don't know which organisms first experimented with this risky strategy, or what immediate advantages they saw. What we do know is that nearly every complex organism alive today, including you, depends on it. Every tree, every mushroom, every animal line can trace its lineage back through countless cycles of chromosome pairing, recombination, and fusion. Sex did not appear as an afterthought. It became a fundamental pattern in complex evolution. Even in lineages that have since reverted to asexual modes, remnants of sexual machinery often remain in the genome, like disassembled tools in a workshop. That persistence hints at how transformative the shift was. Before sex, evolution moved on fewer paths. After its appearance, the landscape of possibilities multiplied, not explosively, but steadily, generation after generation. The world was still entirely microscopic. There were no courtship displays, no parental care, no visible drama. Just cells in ancient seas, occasionally merging, exchanging genes and drifting apart again, quietly rewriting the rules of inheritance for everything yet to come. Life for billions of years had gotten by just fine as a solo act. One cell could feed itself, reproduce, repair damage, and adapt. That approach worked spectacularly well, and still does. But in the Proterozoic, some cells began trying something new. After dividing, they didn't always separate. Instead, they stuck together, forming clusters, filaments, and sheets. It was the faint beginning of a monumental shift. multicellularity. This transition didn't happen once. It appears to have evolved independently in many groups, at least a dozen times, likely more. Different lineages of algae, fungi, slime molds, animals, and plants all found their own ways of building multicellular forms. The idea to live as more than one cooperating cell was not rare. What was rare was pushing that cooperation far enough to build integrated, complex bodies. Early multicellularity was probably simple. A cell divided, and its daughters remained attached. A few rounds of this produced a chain or cluster. Some bacterial colonies, such as filamentous cyanobacteria, still do something similar. Even without nuclei or organelles, they can partition labor. Some cells specialize in photosynthesis, others in nitrogen fixation. These are primitive versions of what would later become true tissues. In eukaryotes, multicellularity took new forms. Some groups produced hollow spheres of cells. Others generated layered sheets. Some began to show differences between inner and outer cells, or between anchoring cells and reproductive ones. Once differentiation started, the advantages compounded. One group of cells could focus on capturing light or nutrients. Others could handle reproduction or structural support. As roles diverged, the whole organism gained abilities no single cell could muster alone. But cooperation posed challenges. Cells sharing a body had to communicate, transport nutrients, and coordinate division. They had to prevent rogue cells from overgrowing or taking more than their share of resources. These problems laid the groundwork for the evolution of developmental controls, growth regulation, and eventually immune systems. Mechanisms for telling self from not self, for orchestrating when and where cells grow, and for managing cell death all trace roots back to these early experiments. Some of the earliest clear evidence of eukaryotic multicellularity comes from Proterozoic microfossils, not solitary cells, but consistent arrangements and patterns. Fossils show linear chains, branching filaments, or clusters whose geometry suggests more than accidental clumping. Some specimens resemble modern red algae in size and complexity, with repeated cell patterns that point to controlled growth. Others appear as flattened sheets or mat-like forms. Energy availability was a major enabling factor. With mitochondria providing efficient ATP production and oxygen gradually becoming more accessible, eukaryotic cells could afford the cost of specialization and communication. Larger bodies mean some cells live farther from direct access to the environment. Oxygen and nutrients must be transported internally. Without oxygen and high-yield respiration, such architectures would have been difficult to maintain. Predation may also have nudged evolution toward multicellularity. Once some eukaryotes gained the ability to engulf other cells, single, naked cells became vulnerable. Clustering together, forming protective matrices, or secreting extracellular materials could make ingestion harder. A group of cells is often more difficult to swallow than a lone one. Over time, defensive clumps could evolve into cooperating collectives. As true multicellularity advanced, development became central. Body plans needed to be reproducible, not random. Chemical gradients within clusters help cells determine where they were, outer versus inner, top versus bottom. Regulatory genes turned on or off in response, guiding cells into specific fates. Scaffolding proteins, adhesion molecules, and signaling pathways all evolved or were repurposed to control this process. Many multicellular lineages remain simple, content with filaments, films, or small balls of cells. They gained some advantages: size, shared protection, localized specialization without progressing to elaborate organs or complex behavior. But some lineages pushed further, building more intricate structures, coordinating large numbers of cells, and eventually generating movement, sensing, and feeding as an integrated whole. Natural selection now had a new level to act on. Not just the cell, but the organism. Traits like overall shape, growth form, and reproductive strategy could influence survival and reproduction. Organisms could become habitats for other organisms, host symbionts, evolve predation, or become prey. The web of interactions grew richer. Single-celled life did not disappear. It remained, and still remains, the backbone of the biosphere. But multicellularity expanded the stage. Life could become bigger, more physically complex, and more ecologically interactive. It opened possibilities that would eventually give rise to forests, reefs, insect swarms, and herds of animals. but those were still far in the future. Here, late in the Proterozoic, the earliest multicellular eukaryotes were modest, small, aquatic, and invisible without magnification. They didn't yet swim with muscles or sense with nerves. They simply grew, adhered, and cooperated. In doing so, they took one of evolution's most momentous steps, choosing, in a sense, not to live alone. Toward the close of the Proterozoic, the pattern began to shift. Not abruptly, not with a sudden surge of creatures, but in a way that leaves a clear signal in the rocks. After nearly 2 billion years dominated by microscopic life, the fossil record starts to reveal outlines and shapes that hint at something newly organized. Bodies with form, with symmetry, with differentiated parts. These organisms weren't animals in the modern sense. They had no bones, no articulated jaws, no eyes. But they were larger, more complex, and undeniably multicellular. This slice of time is known as the Ediacaran period, and the creatures that inhabited it are among the strangest and most enigmatic life forms we've ever discovered. Rocks preserving Ediacaran fossils come from a relatively narrow interval, roughly 635 to 541 million years ago, just after the final snowball Earth glaciation. The ice had melted. Oxygen had risen again to higher, though still variable levels. Nutrients flushed into the oceans from freshly weathered rock. The chemistry of seawater changed, opening new ecological zones. In this altered world, unfamiliar organisms began decorating the seafloor. Ediacaran fossils don't look like modern fish, worms, or plants. They were almost all soft-bodied, with no mineral skeletons. Many had broad, flattened, quilt-like bodies pressed against or just above the sediment. Others formed frond-like structures, disc shapes, or multi-lobed forms. Some lay passively on the bottom. Others may have stood upright or swayed slightly with currents. Most of what we know of them comes from impressions preserved in fine-grained sediments, often aided by microbial mats that coated the seafloor. Those mats helped stabilize the mud and facilitated the preservation of these delicate forms. So what we see today are ghostly casts and molds where living tissues once lay. We still don't know exactly what most of these organisms were. Some paleontologists interpret them as early animals, perhaps related to sponges, cnidarians, or even early relatives of bilaterians, organisms with a left and right side. Others argue they represent an entirely separate branch of multicellular life now completely extinct. A few may have been giant single-celled organisms or loosely organized colonies. Some Ediacaran forms display radial or bilateral symmetry. A few appear to have stalks or holdfasts anchoring them to the seafloor. Sizes range widely from a few millimeters to over a meter enormous by Proterozoic standards. Many lack visible mouths, digestive tracts, or obvious feeding structures. How they obtained energy is debated. They might have absorbed dissolved nutrients directly from seawater, grazed on microbial mats, or harbored symbiotic microbes. Evidence for movement is limited, but intriguing. Certain fossil surfaces preserve track-like features and shallow furrows that suggest some Ediacaran organisms slowly crept or slid across the sediment, perhaps in search of better conditions or food sources. These tracks are simple, a few centimeters wide, meandering, but they imply a front and back, and an ability to move in a sustained direction. Whatever their precise identities, the Ediacaran organisms represent a leap in visible complexity. They show that multicellularity had progressed beyond loose clusters into fully formed bodies with consistent architecture, coordinated development, and likely multiple cell types doing different jobs. These weren't theoretical possibilities anymore. They were living experiments, preserved mid-progress in stone. Their world differed markedly from earlier Proterozoic oceans. Oxygen was higher than it had been for much of the eon, though still lower than later levels. The oceans were settling into new circulation patterns after glaciation, enriched with nutrients from intensified weathering. Microbial life still carpeted many surfaces, but now these carpets shared space with larger, structurally intricate organisms. Light, oxygenity, and nutrient availability were stratified in new ways, allowing more varied ecological layering. Ediacaran communities, as we glimpse them in fossil beds, do not seem especially crowded. Many surfaces show a modest assortment of species, lightly spaced across the seafloor with little overlap. Diversity within a single bed is often limited. Each organism seems to occupy its own patch, with few signs of intense competition, predation, or grazing. It was not yet the densely packed, interaction-rich world of later marine ecosystems. Then, around 541 million years ago, they fade. Not instantaneously, but over a relatively short interval in geological terms. Their distinctive quilted and frond-like shapes diminish and disappear from the record. In their place, we begin to see new traces. Burrows snaking through sediment, small mineralized skeletons, scratch marks, and other indications of more active, mobile life. The spatially ordered, mostly sessile Ediacaran biota gave way to a different ecological regime. Why they declined is still unresolved. It may have been due to changing oxygen or nutrient levels, the arrival of new grazers and burrowers that disrupted microbial mats and surface habitats, or shifts in ocean chemistry. It's possible that some Ediacaran lineages gave rise to Cambrian animals, while others died out entirely. The evolutionary links remain murky, but their significance is clear. They mark a transitional stage, when life on Earth began to build large-scale forms with organized structure. The Ediacaran period did not close the book on the Proterozoic, but it did mark the final chapter. By its end, the world looked nothing like the one that existed 2 billion years earlier. The atmosphere, once dominated by methane and devoid of oxygen, now contained enough O2 to support active metabolisms. The oceans, formerly anoxic and iron-rich, had surface waters that could sustain more complex life. Continental crust had thickened and stabilized through repeated collisions and rifting. Old supercontinents like Rodinia had broken apart. Continental fragments were rearranging, modifying currents and climate. Beneath all that, the biosphere had transformed. Life was no longer exclusively microbial. Simple animals were emerging, soft-bodied and often poorly preserved, but increasingly apparent through trace fossils. Burrow networks and sediments tell us that some beings could move purposefully, pushing through mud and disturbing the seafloor fabric. These activities changed how oxygen penetrated sediments and how nutrients cycled small motions with big consequences. For most of the Proterozoic, organisms reacted passively to their surroundings. Now, some were beginning to reshape those surroundings physically. A worm's burrow is modest, but it affects sediment structure, water filtration, and microbial distribution. Such changes cascade through ecosystems. By this stage, the main evolutionary tools of complex life were in place. Sexual reproduction had become established. True multicellularity with specialized cells and regulated development was no longer rare. Symbioses like mitochondria, and in some lineages, chloroplasts, were standard. Cell communication and tissue-level organization were advancing. Bodies could be built to a plan, not just improvised cluster by cluster. Yet the end of the Proterozoic was not marked by a single catastrophic event. There was no asteroid impact layer spiked with rare metals, no sudden global ash blanket. Instead, the boundary into the next eon. The Phanerozoic is somewhat arbitrarily defined, based on the appearance of abundant, diverse animal fossils in the Cambrian that follows. The shift, though gradual, was profound. Fossils just above this boundary record organisms with clear bilateral symmetry, hardened parts, segmentation, and locomotion driven by muscles. Animals began to dig, swim, and prey in earnest. Ecological networks became more complex, with multiple trophic levels and feedbacks. We call the ensuing diversification the Cambrian explosion, but it was built on Proterozoic foundations. Every critical ingredient that made such an explosion possible: oxygen levels above minimal thresholds, energetic eukaryotic cells, sexual reproduction, established multicellularity, developmental gene networks, was developed, tested, and refined during the long Proterozoic eon, When the Proterozoic concluded, the stage wasn't cleared away, it was furnished. Organisms that had persisted at low diversity now stood ready to radiate into new niches. Small tweaks in body plans could suddenly yield major functional differences: improved mobility, new feeding strategies, better defense. Geological and chemical constraints that had previously held evolution to a slow pace were relaxing. Oxygen was high enough to allow sustained activity. Mineralized skeletons opened options for protection and structural support. Segmentation allowed modular bodies with specialized segments. Vision was about to appear, carving out an entirely new kind of interaction with the environment. Even at this boundary, life was not done with its Proterozoic cast. Microbial mats still coated the seafloor in many places. Ediacaran forms may have hung on in some refuges. Oxygen still fluctuated regionally. Evolution still acted incrementally, generation by generation. But the accumulation of innovations had reached a tipping point. No conscious creature witnessed this transformation. There were no eyes to mark the passing of one eon to the next, no minds to record what was changing. We infer it from fossils, from isotopes, from the composition of ancient rocks. In those faint traces, we see a familiar world beginning to take form. The Proterozoic did not finish with a climax. It stopped because it had accomplished enough. It had stabilized the atmosphere, diversified cell types, invented symbiotic mergers, sexual reproduction, and multi-layered cooperation. It had steered Earth from a world of invisible chemistry into one where structure and form began to matter at larger scales. The curtain didn't fall, it rose. We often imagine evolution as a simple upward line, a progression from small to large, from simple microbes to complex humans. But reality is far messier. There was no pre-written script, no built-in direction aiming toward intelligence or culture. What existed was variation, selection, inheritance, and time, plus an inconceivably vast ensemble of microbial pioneers. Everything about us traces back to them, not in a vague metaphorical sense, but literally, down to the molecules in our cells. Our mitochondria are descendants of bacteria that struck up a symbiotic partnership more than 1 billion years ago. Our cell's DNA is copied, repaired, and expressed using systems first honed in ancient microbial lineages. Our metabolic pathways, glycolysis, the citric acid cycle, electron transport, are updated versions of processes those early cells invented in Proterozoic seas. You don't have to go very far back on your evolutionary family tree before you arrive at entirely microbial ancestors. Every eukaryote, every animal, plant, and fungus is part of a lineage that began when one cell swallowed another and never let it go. Every multicellular organism is built on a decision repeated across countless lineages: to cooperate, to coordinate, to share resources, instead of staying separate. The Proterozoic eon ended a long time ago, but its legacy is ongoing. The internal clocks that govern cell division, the membranes that insulate our cells from the outside world, the way we process oxygen and fuel, the way our genomes recombine during reproduction, these are all products of that time. They were molded by organisms that lived in shallow, murky seas before continents took their modern shapes, before jaws, wings, or brains existed. We carry their solutions and their compromises. The genetic code, with its redundancies and quirks, the way DNA is stored and read, the mechanisms cells use to fold proteins, move cargo, and communicate. Even our vulnerabilities, mutations that escape repair, proteins that misfold, age-related accumulation of damage, are built on ancient frameworks refined but never perfected in those long-ago eons. We also inherit their resilience. Those first microbes survived repeated asteroid impacts, toxic atmospheres, nutrient starvation, global glaciations, and wild swings in ocean chemistry. They rewired their metabolisms, swapped genes, invented new ways to capture energy. Some remained almost unchanged for billions of years. Others transformed so thoroughly that they became parts of us. We are not separate from them, or above them. We are a branch on their tree, one branch among many. Even today, if you weighed all life on Earth, microbes would win easily. By number, by diversity, and by ecological reach, they still dominate. They inhabit our skin, our intestines, our soils, our clouds. They help digest our food, synthesize vitamins, and calibrate our immune systems. We are as dependent on them now as we are descended from them. When we look back into the Proterozoic, we're not just reconstructing a distant alien world. We're looking for our own buried origins, not the origin of life in general, but the origin of the particular kind of life we are. Oxygen-breathing, eukaryotic, multicellular beings built from microbial parts. We tend to equate silence with emptiness, but the quiet of the Proterozoic was not a void, it was density. Those ancient oceans were packed with small, slow, patient forms of life. They swarmed at scales we couldn't see. They tested countless biochemical strategies. They failed often, but when something worked, photosynthesis, symbiosis, sex, multicellularity, it was woven into the fabric of biology and kept. We are not exceptions to that continuity. We are its continuation. The same rules that govern microbial evolution in the Proterozoic still operate now. We breathe oxygen because those organisms made it abundant. We metabolize in certain ways because ancient bacteria discovered those energy routes. We inherit traits via chromosomes because early eukaryotes evolved ways to recombine and separate them. We don't live after the Proterozoic so much as we live on top of it, standing on its shoulders, carried forward by its outcomes. So when you look at your hand, or draw a deep breath, or follow a train of thought, remember, these capacities did not appear abruptly in the last moment of Earth's history. They were assembled incrementally by life that had no eyes, no nerves, no awareness. Cells that formed mats, swapped genes, merged, split, and endured in oceans older than any mountain range on Earth. Those cells are still with you, in your mitochondria, in your microbiome, in the chemical instructions written into your genome. You aren't just from Earth, you're from deep time. The microbial architects of the Proterozoic shaped the world long before anything like us appeared. They created the conditions we require to exist. They were here first. They built the stage. And in a very real sense, they invited us, unknowingly, to join the story. That was the Proterozoic. Not a spectacle taught in most classrooms, not the era of towering reptiles or flowering forests, but a span that occupied nearly half of Earth's history, almost 2 billion years. Over that immense interval, nearly everything foundational to modern life was invented or refined. Cells learned to cooperate and specialize. Oxygen became not just a trace gas, but a usable resource. Continents wandered and collided, reshaping ocean basins and climates. Ice engulfed the globe. Twice, and again, microbes adapted and endured. The Proterozoic did not dazzle with variety of large creatures. It built the underlying systems. It was steady, experimental, relentless. It wasn't about what life looked like. It was about what life could do. Without that long, patient groundwork, there would be no vertebrates, no forests, no ecosystems resembling those we know. Every aspect of what we call complex life is an inheritance from that deep, mostly invisible era. We didn't walk onto an empty stage. By the time anything like us arrived, the scripts, props, lighting, and backdrops had all been prepared by countless generations of microscopic actors. Their work is still encoded in the chemistry of ancient rocks and, more directly, in every cell of our bodies. You don't have to travel back billions of years to find the Proterozoic. You carry its legacy with you every moment, in the quiet machinery that keeps you alive.